We are going to do an experiment called as the total leukocyte count. Okay, for that leukocyte count we need the hemocytometer box having the components, that is the WBC pipette and the hemocytometer slide. Okay, and also we are going to need the diluting fluid we are going to use for this experiment, that is called as the WBC diluting fluid. Okay, that is called as a Turk fluid, not a truck fluid but a Turk fluid, TURK. Okay, the spelling is their mistake in their spelling. Okay, so the composition of this dark fluid is has gentle violet, glacial acetic acid, okay, and distilled water. Okay, gentle violet, it gives the stain to the nucleus of the WBCs. The glacial acetic acid, okay, the term glacial means pure. Okay, the glacial acetic acid means it's a pure form of acetic acid is given. Okay, and this acetic acid helps your solution or the blood. We get the lysis of the RBC cell membrane as well as the lysis of the WBC cell membranes, keeping intact the cytoplasm of the WBCs with the nucleus of the WBCs intact. Okay, so which gives our typical feature of our reflectile halo or groundless appearance of the WBCs in examinations under 10x objective lens. Okay, so as I've told you previously, the identifying point of the WBC pipette is that it has a glass capillary end. Okay, the glass capillary end part is divided into the part which is called a stem. Okay, then we have the circular part which is called as your, or a rounded part or bulge part, that is called as the bulb of the pipette. Okay, and we have certain markings. Okay, there are 10 markings before the bulb, that is written as 0 to just not written anything, just lines are given, and then the very center we have the 0.5, and at the very top part it has one marking given, and just after this bulb end, okay, we have 11 marking. At the very center of the bulb we can see there is a white color bead. Okay, this bead is freely movable. As it is freely movable, it indicates that the pipette is completely dry, and also it helps us identifying the pipette. Okay, if it is a white colored pipette bead, then it's a WBC pipette. Okay, sometimes these white colors get phased away after doing multiple experiments and the color becomes a little bit like reddish, so it's not a definitive identification point. Okay, so the markings are the most identifying point of this instrument. Okay, then the other end of this pipette is called as your rubber pipe. Okay, and we have a mouthpiece. Okay, and this is the hemocytometer slide with improved newer counting area. Okay, we have a hemocytometer slide, we have discussed about those hemocytometer slide. We have the lateral platform, we have the uh, this elevated raised area on the either side of the central shiny area. Okay, this is called as the newer counting area. So we have an A-shaped groove or gutter. Okay, on either side the raised platforms are at a height of about 1 by 10 millimeter. So if we place a cover slip at the very top part of this instrument, we are going to get a gap of 1 by 10 millimeter, okay, between the cover slip and the newer counting area, which is used as a newer chamber for our counting. Okay, so for before the first starting of the experiment, what we are going to do, we are going to pour few ml of your dark fluid in a petri dish. Okay, okay, for the next part of our experiment is that we are going to take the blood sample. Okay, we are going to dip the tip of the pipette completely inside the blood solution. Okay, blood volume or the blood sample, sorry, blood sample, so that while drawing or sucking up the blood, no air bubbles or no air gets inside. Okay, so we in this experiment we don't need any air droplets or air bubbles to get inside our capillary. Okay, then we will draw blood up till 0.5 marking. Okay, then we remove the pipette. Okay, hold the pipette in the horizontal position. Okay, then if it is more than 0.5 marking, okay, that is, we'll count from the number of lines from the bottom, 1 2 3 4 5. The fifth marking is actually the 0.5 marking. Okay, then what I'll do, I will try to bring that blood in the capillary to the level of 0.5. Okay, how we'll do, we'll just tap it. Okay, as we tap it, the blood level will come down. Okay, yeah, exactly, it is in the 0.5 marking. Now I will keep it there only. Okay, try to clean my palm. Okay, then I will complete and make sure you have completely cleaned the edges. Okay, make sure you don't tap it on a filter paper or cotton, because those materials will draw the fluid out from the pipette. Okay, so clean it with a, if possible, with a cloth, or if possible with a soft material. Okay, then you do what, clean the sides of the pipette, because there is some amount of blood that is sticking to the glass capillary end, so that might give you higher result. Okay, then you do what, you take the petri dish having the Turk's fluid. Okay, dip your pipette completely inside the fluid volume, so that you are not drawing any undue air. Okay, then you suck the blood up till 11th marking. The very top marking of the of this pipette is the 11th marking. So I have drawn little bit higher than the 11th marking, because taking less than 11th marking is not recommended. Okay, then I will tap it. Okay, if it comes still down, then you again try to draw it. It should not come so much down. Okay, so it has came down a bit lower than 11th marking. I will again draw the fluid. Okay, this time around again I will try to gently tap it. Okay, and I will try to bring the fluid level up till 11th. Okay, it is now at the 11th level. Now I'll do what, I will grab hold of the rubber pipe in between my pinky finger and my ring finger, and keep the pipette in between my palms, and I will try to mix it gently. Okay, taking care not no fluid or no, the pipette doesn't moves here and there, the rubber pipe, and the fluid doesn't comes out from the pipette. Okay, we'll gently mix it for 30 seconds to 1 minute. Okay, so that the ball, that bead that is present inside the bulb, will mix the solution of blood and Turk's fluid. Okay, now at this time I want to make sure one thing, that in this experiment the Turk's fluid that we are using, okay, we have taken the Turk's fluid at the second part. That is, first part I have taken 0.5 parts of blood. Okay, the second part I have taken, I've told you to take the Turk's fluid. Okay, so obviously the fluid that is present inside the stem, that is only the Turk's fluid, so we don't need that for our experiment. We require the blood mixed with the Turk's fluid that is present inside the bulb. So now what I'll do, I will just discard 2 drops of fluid, 1, 2, okay, 3. Okay, so that will make the fluid that is present inside the bulb, it will come towards the tip of the pipette. Okay, now I will proceed with the next part of the experiment, that is charging in a newer slide. So what we'll do, I will clean a newer slide, and I will clean a cover sleeve and put the cover sleeve on top of the newer counting area. Okay, and in such a way there is little bit shiny silvery area been seen from the sides of the cover slip. So this is the area at which I will try to touch the pipette tip. Okay, so that this 1 by 10 millimeter space between the cover slip and the newer counting area would try to draw the fluid that is coming out from the tip of the pipette towards the whole chamber. Okay, so now hold the pipette at certain angle. Okay, if too much fluid has come from the tip, you just discard that. Okay, now next move on to the next part. Okay, next tilt it a bit and let fluid come out. Okay, and let it pass to this gap of 1 by 10 millimeter. Do it very carefully so that the fluid does not overload the chamber. okay, and try to cover the just the chamber. Okay, if it is going and falling on the gutters, then we call this to be overcharging. You will get extra higher result in the experiment. If you are not covering the whole chamber, then we can call it to be the undercharging. We might get lower value, or we might not even get the values that we are needing. Okay, now we will wait for few minutes, let the blood volume or the WBCs get settled inside the chamber, and we will place it under the microscope and try to count the number of WBCs in the WBC4 WBC counting areas, okay, in the number counting areas. Now